Napari is a multi-dimensional image viewer. You can see a screenshot on the right here of the main Napari interface. There's the main viewing area, which in this example has a picture of some fluorescent cell nuclei. But Napari isn't a standalone image viewer. The real power here is that it's written in Python, so you can use it interactively. So Napari handles Dask arrays natively. This is very powerful for multi-dimensional data because it means that we can do lazy loading and processing of image data. If you put your data into a Dask array, Napari will know how to handle that. On top of it, Napari has this plugin system, so it's really easy to extend with additional functionality. The animation that we have playing right now is a demonstration of how Napari and Dask can work together really well. It was put together by Tally Lambert, and it's taking data from a lattice light sheet microscope. The raw data that's being saved from the lattice light sheet microscope is going to be skewed or deformed. So that means there's a few really interesting steps that need to happen here. We need to lazily load the raw data from disk into a Dask array. And then a computation needs to be applied to deskew the raw data, and that's happening with Dask's map blocks. So that turns the skewed and slightly blurry raw data into something that's well aligned, crisp and clear. And this processing is happening entirely on demand and on the fly, just for the time points that are being selected. sgkit is a general purpose toolkit for quantitative and population genetics. Now, this is a field with extremely large datasets. It's not uncommon to see data from hundreds of thousands of individuals with millions of variant markers genome-wide. Now, Dask is used fairly extensively in sgkit. First, it's used for lazy loading of input data. And then Dask array is used in conjunction with Xarray as a data container structure. Handling some of the computational heavy lifting is DaskML, Dask's machine learning library. We see this especially for tasks like principal components analysis. Last, the Dask data frame collection also gets a bit of a workout, although because this is in connection only with specific file formats, it's technically optional. Tom White recently gave this great short talk at the Dask Summit in May this year, so if you're interested in diving deeper into this topic, I really recommend you check it out. Squidpy is a library that does analysis and visualization of spatial molecular data. One really common application here are studies into cell differentiation. Because changes in the RNA drive the differentiation process, single-cell analysis is really well matched to this type of problem. Dask Image is a specialist image processing library. It's built on top of Dask and SciPy to provide image-specific functionality. Michael, the author of this pull request, has put together this example using the new functionality in SquidPy, and shown here on the right is the result of a watershed operation. Watershedding is a really common image processing task, which is used to split segmented objects that are touching one another. Really early benchmarking of performance for this latest change showed a factor of 2 speed improvement for this particular example. So that's a pretty solid win, even on this 8-core machine, so that's likely a fairly standard laptop. It means that they've effectively removed this ceiling limit for computation as well, because it's going to be much easier to scale up onto a compute cluster, which could probably provide even bigger performance gains. Xarray supports labeling of arrays with dimensions and coordinates. Having access to good, flexible data structures enables so many other libraries in so many other areas of science. It doesn't matter if they're working in very different scientific fields, very different spaces with data that looks and feels very different from one another. Because Xarray is really quite a flexible concept, it works well in all of these different situations. Here are a couple of more typical examples that you might see with Xarray. If you're interested in geospatial data in Dask, I really recommend checking out the Pangeo project and website for more examples and more details there. If you're using Xarray, Dask is an entirely optional extra. But if you do want to take advantage of that, then what happens is that the Dask array collection is used as a drop-in replacement to chunk up these multi-dimensional arrays.